Okay, moving on to Chapter 12, Microbial Control. We're starting to get in a little bit more application type material here, and I don't like to give the impression that we always want to kill bacteria, but for the next few weeks we are focusing on how to get rid of unwanted microorganisms, which is exactly what we were focusing on lab this week, so that kind of fits nicely. Okay, one of my favorite cartoons there. Okay, so historically we've had, lots of, because we didn't understand what caused disease, we've had lots of crazy ways that we've tried to treat disease. The whole leech thing was popular for a long time. It was thought that we needed to get rid of bad blood. That persisted for a long time. Bathing in urine was one thing that I read about. Here's personal protective equipment during 1400s during plague years. Doctors would dress up in these robes and put this weird beak on that had herbs in it so they didn't have to smell the bad air. They often carried a stick so they didn't have to touch their patients. Strapping a live chicken onto your buboes from bubonic plague was thought to perhaps cure the infection. So a lot of crazy things. We've come a long way. So first we're going to start out with some terminology and thinking about how do we choose the best way to try to kill microbes on whatever surface you're trying to clean, or whether it's living or non-living. What sorts of things do we have to think about? Do we use chemicals? Do we use other methods? So we're going to talk through a lot of different physical control treatments. Using heat and radiation are probably the big two. And then we'll go through about 5 or 6 different chemical control treatments as well. All right, so some terminology. It seems like we could just kind of go with this figure from your book here. The difference between disinfection and antisepsis is basically what surface you're treating. So if you are, if you'll see here, and any disinfection is going to be on a non-living surface. So use a disinfectant on a non-living surface. And then an antisepsis is going to be on some living tissue, skin most commonly. What else? So this term sanitation versus sterilization, if you're sterilizing some surface, then you're completely getting rid of all microbes, including endospores. That's a really high bar. Oftentimes chemicals are not going to sterilize. There's a few exceptions, but usually we need some kind of heat to do that, most commonly. And sanitizing is just reducing the microbial load. Oftentimes we use this term when we're in some public health setting. There might be an additional cleaning step that has to take place to follow public health code. So for instance, if you have ever worked in a restaurant and had to wash the dishes, at home, we just soap it up and that's good. But to meet public health standards, there has to be an additional sanitizing step. You have to dip the dishes into a dilute bleach solution after you wash them. Okay, so again, sometimes when you use that term sanitation, it's referring to, some public standard and not just in your home. But sometimes we use it, as just kind of reducing microbial numbers as well. I know. This was a term I wanted to for use on fomites, for use on living tissue. A fomite is just an inanimate object. Usually we use that term to describe, how an infection can pass, through some inanimate object. So a keyboard would be a fomite. You go into the computer room and you type something up right after somebody that was in there was sick, and then you rub your eye, and now, suddenly you're infected, even though you weren't anywhere near somebody that had an infection. So a keyboard, a toy at daycare, that sort of thing, those are fomites that can, again, transmit some kind of infection. The last term here, degerming, this is a term that we use that denotes some kind of physical scrubbing. So if you're going to wash your hands correctly, you don't just put the soap on and then, run your hands under the water. Ideally, you're going to rub your hands together. There's that physical scrubbing action that's important to basically just physically remove the microbes. All right, so if we were in class, I might make you try to think of some of these things, but as it is, you can kind of think about yourself a little bit and then see if you thought of any of the same things that I want to talk about. One thing I forgot to mention here, I wanted to, two suffixes, static and cidal. I wanted to make sure you know the difference between those two terms. If something is, say, bacterial static, that means it just would keep bacteria from growing. We'll say keep microbes from growing. Okay, so the best example I can think of that is when we put our bacteria cultures in the refrigerator in lab until students come back. Okay, that just keeps them from growing. We don't want them to overgrow. Something that's cidal, like fungicidal or sporicidal, that means it kills. Okay, kills the fungus or kills the spores so that they can't germinate again. Okay. All right, so things that we need to think about when we're choosing what method to get rid of microbes. I guess the first thing that I think of is the site to be treated. Okay, is it living? Is it non-living? Does it matter if that site is stained? Does it matter if there's some residue left over? All those things we have to think about. Exposure time. Most of the time, any treatment that we have is going to need some contact time. Even chemicals don't work right away. If you read a lot of household chemicals that you might use to disinfect, on the label it says, kills 99.9% of microbes after 30 seconds of exposure. And I know a lot of times I'm guilty of just spraying it on and wiping it off again. But we do have to consider, can you allow some exposure time? Is that practical? In terms of chemicals, what concentration do we need? Usually if it's less concentrated, it's not going to be as effective, but it is going to be cheaper when it's more dilute. So sometimes there's a trade-off there. Are the cells that you're trying to kill, are they actively growing? We've talked about this already, but cells, when they're in the log phase, at least bacteria, when they're in the log phase of growth, they're generally a lot more susceptible to any kind of antimicrobial treatment. What's the temperature? What's the pH? If you look at this graph here, I'm not sure what microbe it's talking about here, but and exactly, what chemical is applied here, but you can see that the bacteria die much more quickly. 45 degrees Celsius, that's higher than body temperature. That's pretty hot. the chemical works more quickly in a warm temperature than at room temperature. You can see, again, they do die, but just takes longer. Here's the pH. Chlorine is one of the chemicals that we'll talk about. There's lots of different ways to use chlorine, but if you've ever taken care of a swimming pool, you know that you have to pay particular attention not just to how much chlorine you add, but to what the pH is, because chlorine actually disassociates and disappears when you get to a little bit higher pH. So you have to pay attention to that. And one other thing you have to pay attention to is there organic matter present? And what do I mean by organic matter? It could be dirt, it could be vomit, it could be blood, it could be feces. Okay, the tendency is that if you have a big mess like that to clean up, maybe throw some bleach on it and wipe it up. But a lot of chemicals don't penetrate well through organic matter. So really what we need to do is clean it up first, and then if it's the floor that you're trying to disinfect, then add the bleach. So again, that's just, if you are using a chemical to clean your hands that doesn't penetrate through dirt very well, then your hands aren't actually going to be very clean. So usually clean off that organic matter and then disinfect or then do the antisepsis, and it will typically work much better. All right, so in terms of what kind of antimicrobial agent to use, we have to think about what kind of microbe we're trying to kill. So this is not a chart to memorize, but because there's probably a lot of exceptions to this, but in general, the hardest microbe to kill are going to be the prions. And hopefully you look at this list and hopefully you think about, wow, we've talked about all these things. I know what all these things are. I've learned a lot already this semester. But prions, remember, they are infectious proteins. And I think I mentioned in that lecture that they're really difficult to kill. I think, 900 degrees Fahrenheit for 4 hours to deactivate. There are a few really high-level disinfectants that maybe if you, chemicals or enzymes that if you expose them to prions for like 10 hours, they may deactivate the prions, but they're very difficult to get rid of. And then, of course, endospores, we've talked about how difficult they are to kill. They survive longer exposures to radiation, for example, longer exposures to high temperatures. And next one here, cysts of Cryptosporidium. So this is a protozoan. We talked about how some protozoa have this kind of dormant cyst stage. And these are not as difficult to kill as bacterial endospores, but they do sometimes, as they found in Milwaukee in 1993, they can survive sometimes application of chlorine. So it's kind of an interesting example. It's I'll just show you this just a second. Let's see, what are my numbers on this? In 1993 in Milwaukee, they had a Cryptosporidium outbreak in their tap water. Okay, so it's not clear like how that happened all of a sudden. They ended up kind of replacing a bunch of filters and it got rid of the problem. But 400,000 people were sick with diarrhea from ingesting Cryptosporidium. So imagine that. That would be tough. So out of a city of about 1.5 million people, 400,000 were sick. So it was pretty widespread. At least 69 people died from it, probably people that were kind of medically fragile already. So, and interestingly enough, To begin with, people were told to, drink water. When you have diarrhea, you should drink lots of water. And obviously, when they were drinking their tap water, that made it even worse. So hopefully that doesn't happen again. That would be bad. Kind of on the lower end of the scale here, I think we talked about this, that envelope viruses are usually the wimpiest ones in the environment. Okay, because they have that membrane dries out really quickly. And if the membrane dries out, then the virus is not able to infect cells anymore. So things like influenza, COVID, they're pretty wimpy in the environment. And here I think there's probably some variation here. Gram-positive bacteria, that peptidoglycan actually absorbs disinfectants a little bit more readily than a gram-negative bacteria. Large non-enveloped viruses, here the small ones are way up here. This is like norovirus. It's a small non-enveloped virus. And I mentioned how that one persists for so long in the environment. Okay, Mycobacterium, of course, remember that's our acid-fast organism, so they have a little tougher cell wall, that waxy cell wall. You would expect them to persist a little bit better than maybe other kinds of regular cell-walled bacteria. Okay, so that's kind of interesting to think about. But again, don't memorize, think about what's the hardest to kill and what's the easiest to kill. When possible, we're going to talk about how these antimicrobial agents work, kind of the basic mechanism. And there's a lot of these antimicrobial agents that they just kind of do everything. But two basic mechanisms, disrupting the cell wall and cell membrane. Obviously, if the cell wall is disrupted, then it's going to be a little bit more susceptible to osmotic changes. It's more susceptible to bursting. We talked about that cell membrane, how important it is to have an intact cell membrane when the cell is trying to get ATP from that electron transport chain. It has to keep the proton gradient intact there. And just, when the cell membrane is disrupted, things are going to leave the cell that aren't supposed to leave. Things are going to enter that aren't supposed to enter. And of course, envelope viruses have to have that in order to be able to attach to a host. Disrupting, we'll say proteins and DNA, another kind of big category there. Obviously, proteins have to hold their particular shape, and that's crucial for the function of them. If some of those bonds are broken, they're not going to be functional. And we've already talked about mutations altering nucleic acids as well. All right, the first, I just wanted to have a slide where I talked about the methods of physical control that we're going to talk about and the chemical agents that we're going to talk about, because there are other ones that are mentioned in your text. So these are the ones that you'll be responsible for, and we'll work our way through here. Heat is kind of the first thing that you think about when you're trying to kill cells. So we'll explore that first. In today's lab, exercise 12 dealt with heat, so there's a little overlap here from what your lab instructor will tell you. But heat basically causes all different kinds of problems for the cell, kind of destroys everything. There's some cells are a lot more tolerant to high temperatures than others, so we like to try to measure that tolerance. One way is by trying to measure the thermal death time. And that's one of the things that we're going to try to measure in lab. But that's defined as the shortest time that kills an organism at a given temperature. Okay, so we're going to try to maybe make a data table similar to this. Here's a given temperature, 65 degrees Celsius, And what you're going to do in lab is expose your cells to that temperature for a short period of time, and then take an aliquot out and put it on an agar plate, and we'll see if that grows up later. So we always do a control and make sure it's viable before we expose the organism to heat. And then say we got this data. So if we were trying to understand the thermal death time, the shortest time that kills at a given temperature, for E. coli and Staph aureus, the thermal death time would be 4 minutes. For Bacillus, the best we could say would be greater than 10 minutes. Okay, remember what's special about Bacillus, it's a spore former, so that's not surprising. We hope to see that in lab, that it can survive a lot longer. Again, these are all mesophiles, and if we kind of, these short exposures to high heat, that's what we're testing. Sometimes we test it a little bit different way, and we try to test the thermal death point. Here we're just changing the variables around a little bit. That's the lowest temperature that kills at a given time. This is a little bit more difficult to test because you have to have lots of different water baths at different temperatures. Usually we designate the temperature as 10 minutes. So we expose our organism, and then, again, put an aliquot on a agar plate and see if they survive. So here, the thermal death point for E. coli would be 60. It's, again, the lowest temperature that kills. For Staph aureus, with this data I made up, it would be 65. And for Bacillus, again, greater than 70 degrees Celsius. Okay, it's a little bit more what we would say thermoduric, meaning it can withstand high temperatures for short periods of time. Not a thermophile, it doesn't grow optimally there, but it can withstand it. So that's a term, thermoduric, that your lab instructor might mention. It's just the ability to withstand high temperatures for a short period of time. All right, so let me just start our talking about moist heat applications just a little bit here. Usually we're going to use some kind of moist heat if possible, because moist heat is just going to penetrate much more quickly, so we don't have to use as high temperatures, and it just is a little bit faster. So the first thing you think of if you're trying to disinfect something maybe at home is to drop it in some boiling water. Okay, so that water is going to be 100 degrees Celsius. When it gets higher than that, it's going to turn into steam, right? So if you have something like a pacifier or something like that you want to disinfect, you could drop it in some boiling water and leave it in there for a few minutes, and that would do the trick. It is considered a disinfection because it kills vegetative cells within 10 minutes. Spores, interestingly enough, take at least 30 minutes, greater than or at least 30 minutes to kill. So that's kind of surprising to me that they would withstand boiling water that well, but I've read that in many different places. Okay, so boiling is not considered a sterilization. It will kill everything except spores, so we'll kill vegetative cells really well. Another very common application of using moist heat is pasteurization. Okay, so that's named for Louis Pasteur, who first heated up grape juice in order to kill the bacteria that were spoiling the wine. This is also a sanitation. We are not boiling, most commonly we pasteurize milk today. We're not boiling the milk usually. It's just going to, at least sanitation as it's done in the US, is just heating up that milk just enough to kill pathogens, so killing disease-causing organisms. It might not kill thermophiles, but that's okay. We're not worried about them. They don't cause infection. Okay, so this is going to reduce microbial numbers to safe levels, meaning it's going to kill the pathogens, And, a big thing, it's going to increase the shelf life of the milk. Okay, so you cut down on those microbial numbers. That's routinely tested and make sure pasteurization is reducing microbial numbers, and then it takes them a lot longer to reproduce to an extent where the milk would not be safe to drink. Interestingly enough, this link here is to one of my favorite commercials. It came out a few years ago. It showed a family drinking chunky milk and chewing milk. Interestingly enough, your milk, if your milk is chunky, that's not necessarily going to make you sick. It takes a long time for milk to go bad to the extent that it would make you sick. It might not taste good, but you can definitely use milk past the due date. So I do that. Well, usually we drink it up, but I do that all the time, and it's good for a while. So here's a figure in your book that talks a little bit about pasteurization. This is the form that we do most commonly do pasteurization in this country. They call it high temperature short time. That's also known as flash pasteurization. Okay, again, this is the most common in the U.S. In other countries, it's more common to do this ultra high temperature pasteurization, where you heat the milk up really, really hot and really, briefly, but it's enough to sterilize the milk. So then it can be put out, in the grocery store at room temperature. So that really cuts down on the expense of transporting milk. The big problem, and the reason this hasn't caught on in the U.S. is because the taste is altered a little bit. So we see that more in Europe. You can buy milk that way here. I haven't tried it, but apparently it tastes a little bit different. And just, why do we pasteurize milk? Before we did that, and, worldwide, milk is still the most common cause of foodborne illness. But in this country, even, before we pasteurized, there was all sorts of infection that were passed through milk. Today, is it safe to drink milk that hasn't been pasteurized in the U.S.? According to the CDC, yes, it is safe to drink raw milk, but you shouldn't drink raw milk if you are immunocompromised or if you're pregnant. Everyone else it's safe for. Personally, I wouldn't. But, in this country, our cows are very healthy. Farmers want to keep their cows healthy so they produce more milk. They've got their vaccinations. We test milk all the time, both raw and after it's been pasteurized. But because cows are so healthy that it is considered safe. Pasteurization does cut down slightly on a couple of the vitamin contents. If you go online, there's a thriving community of people that say we shouldn't be pasteurizing milk and that raw milk is much healthier. But yeah, personally, I'm gonna stick with pasteurized milk. Okay, and lastly, our last application of moist heats, and then I'll stop for today. The autoclave. Okay, this is a good picture of what an autoclave looks like. They're big, double-walled metal devices. They are usually dirty, like this one in the picture, so that's why I showed that. These are devices that use steam under pressure in order to sterilize. Okay. And usually the parameters that we bring our autoclave up to, so whatever you're trying to sterilize, whether it's media in the microbiology lab or swabs or whatever, whatever, anything, we bring it up to 121 degrees Celsius at 15 pounds per square inch of pressure. So that's an additional atmospheric pressure. Atmospheric pressure is 15 pounds per square inch. We're adding an additional atmospheric pressure for 15 minutes. So how do we get water up beyond boiling point? It's because we've added the pressure. And when you increase the pressure, that raises the boiling point. So we can kind of superheat the water, and again, it acts really, really quickly, that superheated water as well as steam, and that's going to sterilize, kill even endospores. Okay, it does have the disadvantage, obviously, you can only use it on things that are heat resistant, but anything that you can get wet, again, we use this for everything in the lab, glassware, everything that we want to sterilize. Things like oils and powders, you can't autoclave. You can't autoclave thermometers because they would burst. Some kinds of medical instruments, you might dull them a little bit. But again, we use this all the time. We will put autoclave tape on there. So anything, say we've got some media that we want to autoclave, we put some tape on there before we put it in. And then as long as the autoclave has come up to the appropriate temperature, there's some, lines on there that will turn black. And that's how we know that the autoclave is working properly. Because the autoclave do break down pretty often. And so we need to know if things are not actually sterile. Okay, I will continue with dry heat on lecture on Monday. I've talked a little bit longer than I meant to. Sorry about that. I hope you have a good weekend and stay safe.